Hello, good morning everyone. Today we are going to learn what is IVIVC, in vitro in vivo correlation, and what are the requirements, what exactly is a bioequivalence study. This section is done by Prashant Naik, myself. I'm going to take you deep insight on IVIVC and bioequivalence studies. What is IVIVC? In vitro in vivo correlation is a predictive mathematical model describing the relation between in vitro properties like dissolution rate and in vivo responses like plasma drug concentration. It helps determine whether dissolution test can replace in vitro studies or in vivo studies. Objectives of IVIVC: ensure batch to batch consistency in the manufacturing of tablets or any other pharmaceutical preparation, aid in new dosage form development. Whenever we do a new dosage form, IVIVC is very important. Help set meaningful dissolution specification. Approaches are two types. One is called as direct correlation. We correlate the data of in vivo and in vitro and then predict on based on a mathematical model. Or one more method is there which is called modified dissolution. Types of quantitative correlation. Plasma level data relates percentage drug dissolved with percentage absorbed. That is, you can have a Cmax, Tmax, etc. AUC. So this all is told about a plasma drug level, how quantitatively we can correlate. Urinary excretion, correlation drug dissolution with urinary drug excretion. Pharmacological response links dissolution data with effects like LD50. Statistical movements using mean resolution time versus mean residence time. Limitations are factors like poor resolution, methodology, particle size, or pre-systemic metabolism can be weaken or prevent correlation. What is levels of IVIVC? There are four levels. One is called as point-to-point correlation, most reliable dissolution profile, can replace in vivo studies for change in formulation or manufacturing. Level B uses mean values, not point to point, less reliable for regulatory purpose. Level C, single point correlation, that is P50% versus AUC, or Cmax, useful only as a guide. Multiple levels correlation. C correlates multiple points to multiplex PK parameter, pharmacokinetic parameters. Now we'll understand how these are being class of drugs, what is the solubility and permeability parameter, and then how it affects the IVIVC. We have seen IVIVC A, B, C. We'll correlate it with class of drugs. Class 1A, high and site independent, solubility is high and site independent, permeability is also high. So what is IVIVC correlation expected over here is of level A. This has to be remembered. In case of 1B class, high and site independent depending on neural absorption window. So it is level C is expected. Level class 2A, low solubility and high permeability. Low and site independent. So IMIC level will always be expected as A. 2B, low and site independent, dependent on site and narrow absorption window, little or no IVIVC. Class 5th A acidic, class 5th B basic, class 4th A 5th A is variable and permeability also variable, little or no IVIVC. Class VB basic, variable in case of solubility and permeability, and IVIVC level is expected to be A. Now, BCS based biowaivers or in vivo bioavailability and bioequivalence studies. What exactly is this? According to BCS, in vivo bioavailability and bioequivalence studies need not to be conjugated for drug products. Under following circumstances, rapid and similar dissolution, high solubility will be there for some drugs, high permeability can also be the case, and wide range of therapeutic index can be seen. Excipients used in dosage form are same as those present in the approved drug product. The key terms in the drug product comparison equivalency. A general term for comparing drug product based on specific characteristics or standards. Now, what is chemical equivalence in this? They are called as equivalents. What are chemical equivalents? Products contain same active ingredients in the same amount. Whenever we are found in any kind of two drugs, they are said to be chemically equivalent. What is pharmaceutical equivalence? Products have the same strength, purity, quality, and dissolution, but differ in their excipients. So, the two products differ in the excipients. We always say it is pharmaceutical equivalence. Now, what is bioequivalence? Bioequivalence products shows no significant difference in rate and extent of drug absorption. Plasma concentration time profile are similar. Bioequivalence statistical significance differences in bioavailability. Therapeutic equivalence are products which are having same clinical safety as well as efficacy profile. Types of bioequivalence studies. Bioequivalence can be demonstrated as in vivo or in vitro. In vivo bioequivalence studies. Following sequence of criteria is meaningful in expressing the need of in vivo studies. Oral immediate release products with synthetic action indicated for serious conditions requiring an assumed response. Narrow therapeutic margin it should be. Pharmacokinetic complicated by absorption less than 70% or absorption window. Non-linear pharmacokinetics. Pre-systematic elimination should be greater than 70%. Unfavorable physiological properties, low like slow solubility, what happens, metastable modification, instability, etc. Non-oral immediate release products. So what are the non-oral immediate release products? Injections. These all are having problems in bioavailability. Modified release products with systemic action. In vivo bioequivalence studies are conducted in the usual manner as discussed for bioavailability studies, that is, the pharmacokinetic and pharmacodynamic methods. In vitro bioequivalence studies is none of the above criteria is applicable. Comparative in vitro dissolution studies will suffix in vitro studies, that is, dissolution studies, can be used in lieu of an in vivo bioequivalence under certain circumstances are called as biowaiver exemptions. Biowaivers, the drug product differ only in the strength of the active substance it contains or it provides all the following conditions. Pharmacokinetics are linear, qualitative composition is the same, ratio between active substance and the excipient is the same, or the ratio between excipient is the same. Both products are produced by the same manufacturer at the same production site. Bioavailability or bioequivalence study has been performed with the original product. Under the same test condition, the in vitro dissolution rate is the same. The drug product has been slightly reformulated or the manufacturing method has been slightly modified by the manufacturer in ways that can convincingly be argued to be irrelevant sorry to the irrelevant for the viability. Third point is the drug product meets the following requirement. The product is in the form of solution or solubilized form, elixir, syrups, tincture, etc. Product contains active ingredients in the same concentration as the approved drug product. The product contains no excipient known to significantly affect absorption of the active ingredients. Fourth point is an acceptable IV/IVC and the in vitro dissolution rate of the new product is equivalent with that of the already approved medical product. Moreover, the product is intended for topical application like creams, ointment, gels for local effect. The product is for oral administration but not intended to be absorbed. Antacid, radio-opaque substances. Product is administered by inhalation. inhalation or a gas or vapor. Criteria for drug products listed above indicate that bioavailability and bioequivalence are self-evident. Now, what is bioequivalence experiment study design? There are various types of tests that are usually employed in clinical trial: bioavailability and bioequivalence. What are randomized designs? If in a completely randomized design, all treatment favors level are randomly allocated among experimental subjects. Method of randomization: label all the subjects with the same number of digits. For example, if there are 20 subjects, name them as 1 to 20. Randomly select non-repeating numbers like simple randomization with among these labels for the first treatment and then repeat for all other treatments. Advantages of this method is the design is extremely easy to conduct. It can accommodate any number of treatment and subjects. The design is simple and easy to analyze even though the samples might not be in the same for each treatment. Disadvantages: although the design can be used for a number of treatments, it is best suited for situations in which there are relatively few treatments. All subjects must be homogeneous as possible. Any extraneous source of variability will tend to inflate the random error term, making it difficult to detect differences among the treatment mean responses. What is randomized block designs? First, subjects are sorted into homogeneous groups called blocks, and the treatments are then assigned at random within the blocks. Method of randomization: subjects having similar characteristics are formed as blocks, and the treatment are randomized with each block. Yeah, what are the advantages? The design is extremely easy to conduct. It can accommodate any number of treatment and subjects. Design is easy and simple to analyze even though the sample size may not be the same for each treatment. Disadvantages of this: although the design can be used for any number of treatment, it is best suited for situation in which there are relatively few treatments. All subjects must be as homogeneous as possible. Any extraneous source of variability will tend to inflate the random error term, making it difficult to detect the differences among the treatment or factor level mean response. What are randomized block designs? Subjects are sorted into homogeneous groups called blocks, and the treatment are then assigned at random within the blocks. What exactly is the method? Subjects having similar background characteristics are formed as blocks. For diabetic hypertension, separate blocks have been characterized. Treatment are randomized with each block, just like simple randomization. Randomization for different blocks are done independent of each other. Advantage of this method: with effective and systemic way of grouping, it can provide substantial more precise results than a completely randomized design of comparable size. It can accommodate any number of treatment and replications. Different treatments need not have equal sample value. Statistical analysis is relatively simple. The design is easy to construct. If an entire treatment of block need to be dropped from the analysis for some reason, such as spoiled results, analysis is not thereby complicated. Variability in experimental units can be deliberately introduced to widen the range of validity of the experiment results without sacrificing the precision of results. What are disadvantages? Missing observation within a block require more complex analysis. Degree of freedom of experimental error are not as large as with completely randomized design. Now, repeated measures, crossover, and carryover designs. It is essential a randomized block design in which some subject serves as block. The same subject is utilized for each of the treatment under the study. Since we repeatedly measure each subject, we get the design named repetitive measures. The study may involve several treatments or a single treatment evaluated at different point of time. Administration of two or more treatment, one after the another in specified or random order to the same group of patients is called crossover design or changeover design. Drawback of crossover studies is the potential for distorting due to carryover, that is residual effects for preceding treatment. To prevent carryover effects, generally about 10 elimination half-lives are required. Clinical trials to monitor safety and side effects. Method of randomization. Complete randomization is usually randomize the order treatment for each subject. Randomization for different subjects are independent of each other. Advantage, they provide good precision for comparing treatments based on source of variability between subjects are excluded from the experimental error. It is economic on the subjects, particularly important when only few subjects can be utilized for the experiment. When the interest is in the effect of one treatment over time, it is usually desirable to observe the same subject at different points rather than covering different subjects at the specified time. Now let us study what is the Latin square design. Completely randomized design, randomized block, repetitive measure are experimental, whereas person subject volunteers remains on treatment from the start of the experiment till the end and thus are called as a continuous trial. In Latin square, however, each subject receives each treatment during the course of experiment. A Latin square design is a two-factor design, subject and treatment and the two factors, which one observation is each cell. Such as design is useful compared to the earlier ones when three or more treatments are to be compared and carryover effects are balanced. In a Latin square design, rows represent subject and column represents treatment. A RXR Latin square design is a square with R rows and R columns, such that each of the R square cells contain one and only one of the R letter representing the treatment, and each letter appears once and only once in every row and every column. Latin square is called standard if the first row and the first column consist of R letter in alphabetical order. Randomized balanced crossover Latin design are commonly used in bioequivalence studies. Now, what are the advantages of this method? It minimizes the intersubject variability in plasma drug level, minimizes the carryover effects with which could occur when a given dosage form influences the bioavailability of a subject of a subsequently administered product, intra-subject variability, minimize the variation due to time effect. Treatment effects can be studied from a small scale experiment. This is particularly helpful in preliminary or pilot studies. Moreover, it is possible to focus more on formulation variables, which is the key to success for any bioequivalence studies. Disadvantage, the use of Latin square design will lead to a very small number of degree of freedom for experimental error. Only a few treatments are studied. On the other hand, many treatments are studied. Degree of freedom for experimental error may be larger than necessary. Randomized required somewhat more complex than that for the earlier design considered. Study takes a long time since an appropriate washout period between the two administration is essential, which may be very long if the drug has a long T half. So today what we have learnt is what is in vivo in vitro correlation. So your takeaway will be IVIVC, levels of IVC, level A, B, C, we have learned. Then what are the various designs of viability study? What is a randomized design? What are clinical trials? What is a Latin square design? So thank you for now. We'll meet in the next class. Have a nice day. Bye.